Susan Zielinski, are you with us this morning?
Yes.
Hi, good morning to you.
Good morning.
Thank you for your part in helping arrange our morning session, and just want to make sure that my co-captain was on the call with me. Susan Zielinski from Janssen Johnson is a MOBIO board member, has been for a very long time, and for all of the great programs that we've done at MOBIO, one of the strongest programs we ever developed was our biobenchmarking legislative education tour. And Susan was in that very core group of 3 or 4 people that had the vision for that, bought into it, worked behind the scenes for 1.5 years before we even announced it, and rolled out a really strong program. And Susan, thanks for being our partner at MOBIO, all your leadership inside the Missouri State Capitol on life science issues, and for helping to arrange today's program. Okay. We've got 31 participants. Looks like we're climbing up to 33. I'll keep watching the participant. And can people see me?
Yep.
Super. I want you to remind yourself to please mute your call during the session. And Dr. Nettles, are you on with us this morning?
Yes, I am.
Super. Good morning to you.
Good morning.
It's my pleasure to introduce Dr. Nettles to the MOBIO Breakthrough session. Dr. Nettles is the Vice President for Medical Affairs for Janssen Infectious Diseases, which is a subsidiary of Johnson & Johnson. In his capacity of leading that U.S. infectious diseases team, he enables Janssen to bring critical market-leading therapies to people living with HIV. He and his team also work closely with global R&D colleagues in support of research that spans both vaccines and therapeutics. Rick has more than 15 years of experience in the pharmaceutical industry. Prior to joining Johnson & Johnson in 2011, Dr. Nettles held various positions with Bristol Myers Squibb, including R&D leadership roles in virology for the company's discovery medicine, clinical pharmacology, and global clinical research organizations. Dr. Nettles holds a Bachelor of Science degree from the University of Notre Dame, and after receiving his medical degree from Duke University, Rick completed his internal medicine residency at Duke University Medical Center and his infectious diseases fellowship from Johns Hopkins University. He served as an assistant professor of medicine in the infectious diseases department at Johns Hopkins, where he also attended in the Johns Hopkins HIV clinic. And I had the pleasure and benefit of hearing Dr. Nettles speak to the Nebraska Pharmacy Association a couple weeks back, another program that was very well received, answered a lot of questions by their members, and we're delighted to have this opportunity to hear from Dr. Nettles on the work they've been doing at an unprecedented pace, and along with probably a full bandwidth of other projects in terms of medical development for Johnson & Johnson and for Janssen that did not go away just because we decided to have a pandemic in 2020. So I know his plate is full. We're grateful for his time this morning, and we just have really strong attendance today. Dr. Nettles, thank you for being with us, and apologize for not being able to greet you ahead of time this morning, but we're going to flow through this and take care of each other as best we can.
Absolutely. Thank you very much for the kind introduction and the nice words. I'm glad that you caught this presentation one time before. I'm going to try to share my screen, so let's see how that goes. And then you'll be able to see my slides.
Okay.
Can you see those slides now?
Yes, we can, or I can.
Okay, outstanding. So I want to, do you want me to go ahead now and then we can take questions at the end? Does that sound good?
That'd be perfect. Thank you.
Okay, wonderful. Well, thank you very much for inviting Johnson & Johnson to speak today. I'm speaking on behalf of the company. I'm speaking on behalf of Janssen, which you might know is the pharmaceutical division of Johnson & Johnson. And with regard to J&J in general, we feel very passionately that we have a responsibility to step up during times like these. J&J has a history of coming to the aid of global and local entities during times of disaster, whether they be natural disasters, hurricanes and such, or health emergencies such as the pandemic we're facing right now. We feel it's our responsibility because as a 130-year-old company, we really have a history and connections across the world. With 130,000 employees, we have a global reach, and we feel that we're in a unique position to be able to bring solutions during times of crisis like these. So what I'd like to do today is to talk to you about what J&J is doing in response to the coronavirus pandemic. And for the majority of the time, what I'll be doing is describing what we're doing with regard to our vaccine strategy. And then I'll briefly touch on our quest to find therapeutics to treat those who have become infected with the coronavirus. This slide lays out our timeline over the past year of what we have been working on, particularly around the vaccine. So as you know, reports started to come out of China in December of 2019 of this new infection. The Chinese government released the genetic sequence for the coronavirus in January of this year, and that's really when we began working on our vaccine. Importantly, part of the genetic sequence of the coronavirus that was released codes the spike protein of the virus, and that piece of genetic material was important to our being able to develop a vaccine. We had been working on vaccines at that time for things like respiratory syncytial virus, Ebola, RSV, Zika, and HIV. We were able to use the platform that we've been working on and plug in the genetic sequence for the coronavirus. So we're able to get a fast start on the development of a vaccine, even in this situation where we had a newly emerging infectious disease. That led to us selecting the lead candidate in March of this year. That's when we selected the candidate we were going to take forward in development. We started preclinical work, and about 2 weeks ago, we published in the journal Nature our preclinical work on non-human primates. So importantly, in that publication, some of the things that we showed was that with the lead candidate that J&J has selected, with a single dose of the vaccine, and that's important because most of the other vaccines that are ahead of J&J or developing in the same time frame as J&J are a 2-dose vaccination series, which obviously adds to logistical complexity. But anyhow, with a single dose in non-human primates, we saw a very nice and robust cellular and antibody response. And that's what you want to see with a vaccine. You want to see that two-pronged response to your vaccine. We also saw in vaccination of the non-human primates that we were able to completely protect the lower respiratory tract of the 6 monkeys that received our lead candidate vaccine. So again, that's important. You vaccinate these monkeys, and then a few weeks down the road, you challenge them by infecting them with coronavirus. And in the monkeys that received our vaccine, we were not able to find any of the virus in the lower lungs of the monkeys. And importantly, we saw an antibody response in these monkeys that was 4-fold higher than monkeys or humans who had been actually infected with the coronavirus. So we're very pleased that our vaccine is working well in non-human primates. That allowed us to move forward into phase 1 human development with our vaccine in July. In that phase 1, phase 2A human clinical trial. We will be studying over 1,000 individuals, including individuals in the age range of 18 to 55. And when shown that the vaccine is safe in that population, extending up over the age of 65 to make sure that the vaccine is safe and tolerated in the elderly. In the event that that clinical trial goes well and the vaccine is shown to have an immunologic response and is safe and well tolerated, we would have interim results of that in September, and that would allow us to move forward into a phase 3 clinical trial in September of this year. Then again, if all goes well in that trial, and that trial we're looking to enroll approximately 30,000 or more individuals, so it's a very large clinical trial. If all goes well, we would then move forward to conversations with the FDA. And if they agreed, we would be able to make the vaccine available under an emergency use authorization in the early part of 2021. I'd like to talk a little bit now about the construct that we're using for a vaccine series. So as I mentioned, one of the ways that we were able to very quickly begin the development of our vaccine is we had been using a vaccine platform to develop vaccination against other serious diseases. And what you actually do with this vaccine is you take a piece of the genetic material of the coronavirus that you're trying to protect against, and you plug that into a different type of a virus called adenovirus, which causes the common cold. You've made that adenovirus into a virus that cannot replicate itself, so it can't make you sick. So the adenovirus serves as a Trojan horse, if you will. You inject the adenovirus into the muscle of the individual you're vaccinating. The coronavirus genetic sequence for the spike protein is expressed, and you actually develop a robust immune response to the coronavirus based on that vaccination. J&J developed multiple candidates using this technique and then selected the candidate that induced the most robust immune response in the non-human primates. And we've selected a candidate that we were able to scale up and produce rapidly and in large amounts. So we have begun to produce our vaccine candidate at risk. In many instances, what you'll do with vaccine development is you'll wait until you have success in your phase 3 clinical trial, and perhaps even success in your discussions with the FDA and have an approved vaccine before you really scale up in large quantities your development.
Tell everybody I said hi.
Folks, can we, if you're not Dr. Nettles right now, could you please mute?
So thank you. So what we've done in this instance is, rather than doing things in serial, one after the other, we're taking some risks with regard to our investment in production capacity. This adenovirus-based platform, we're able to produce very large amounts of our vaccine, and we've built up our capabilities to produce large amounts of vaccine here in the US. J&J feels strongly that no vaccine is effective if no one can afford to get it and if no one has confidence in receiving the vaccine. So we have committed to making our vaccine available publicly for a not-for-profit price during the pandemic emergency. We're committed, as I said, to producing the vaccine in the US and producing a large supply in the US, but also global supply. As you know, we've heard a lot about herd immunity over the last couple months. It's really important to vaccinate all of the world to make sure that we can stop the spread of this virus, rather than just targeting small populations with our vaccine. As you may have heard last week, we came to an agreement with the US federal government to supply them with 100 million doses of the vaccine early next year. Before I stop, I'll just give you a brief overview of what we're doing, looking for solutions for those who are infected with coronavirus already. So a vaccine's role is to prevent an individual from becoming infected, but it's also important to develop treatments for those who have already become infected. We have been collaborating with BARDA, part of the US federal government, to screen our compound library to see if we have any compounds already on hand that might have antiviral activity against the coronavirus. We've started and screened through compounds that are already approved and then moved to those that are in phase 3 development, and then we're moving earlier and earlier. We've even offered to screen compounds from other competitor companies through this collaboration with BARDA. And as we've learned with this COVID infection, it's not just a typical infectious disease. You also see a significant amount of pathology caused by the severe inflammation as a reaction to the virus. So we're also conducting clinical trials looking at our immune modulators to see if perhaps those could be of benefit, particularly for those patients who have the most severe coronavirus infection requiring hospitalization or ICU admission with intubation. Those trials are ongoing. And a couple times I've referred to our partnerships that we've been working on, and with this slide, I just want to say we are sincerely grateful to the partnerships that we have engaged in, whether it be the US government through BARDA or the NIH, in frequent discussions now with the CDC and the FDA, with Beth Israel Deaconess Medical Center, who we're collaborating with on developing the vaccine specifically, the Rega Institute, helping with the screening of the other compounds. And I mentioned the scale up of the ability to produce the vaccine in large quantities. Emergent Biosolutions and Catalent are the US partnerships being able to produce the vaccine and then package it. And there's multiple others, but we just want to say that we feel in times of this big of a health crisis, partnership is the way to go, certainly. We're committed as J&J to work as quickly as we can to find solutions. We feel it's our responsibility to do that. We want you to know that at J&J, we really are working around the clock on the vaccine and the other solutions to get it here as soon as we can. But at the same time, our primary goal is to bring a vaccine which is safe and effective. So it's also important for me to say that we are not cutting any corners. The size of those clinical trials are very large and are the same size as those which would be conducted under usual vaccine development. So it's really this with safety and efficacy as the most important factor, moving as fast as we can under those principles. So I will stop there and take any kind of questions that you guys have.
Dr. Nettles, thank you very much for that overview. I know we'll have some questions from our audience. What is keeping you up at night and worrying about J&J's positioning on this in order to be successful? Do you have needs outside your organization that you're really aggressively going after now to try and hit those main objectives of safety and efficacy?
Yeah, usually probably my answer here would be to say that we need to see the safety and the efficacy in the phase 3 clinical trial, and that is certainly true. We feel good about where we're going with regard to our research and development efforts. Or you would hear me say something about being able to compete with other vaccines that are out there. There's now hundreds of them in development. But the truth is, in this situation, that we need multiple vaccines to succeed, so that we're sure that we have a few that are out there, and that everybody who wants one of the vaccines can get it. So that's less of concern to me in this instance. And the good thing is, on committees like this, often there's other pharmaceutical companies, and we're all saying the same thing. It's a spirit of we need to have multiple vaccines as soon as possible. So what I would say, the biggest thing that has me concerned now is we need to find a distribution system that we can vaccinate the very, very 300 million Americans as fast as we possibly can. And we need to find a way to convince those 300 million Americans that the vaccines are safe, that corners haven't been cut. So those are the things that are keeping me up at night the most. I know the CDC and others are working on those things as fast as they can.
Maybe that will be the role of the NFL this year, is to is to make all attendees get vaccinated as they come in the stadiums, 80,000 at a time.
Yeah, that might work.
I'm going to unmute everyone and see if that will help make sure that people can get their questions asked, or if you'll unmute yourself and ask the question. I know I've seen Samuel Lamb has asked a question. William Folk has. If you guys would like to unmute, please do so.
So this is Bill Folk. Thanks for the very nice talk, Dr. Nettles. SARS and MERS are neurotropic viruses, and so that raises the question of whether an antibody response is going to prevent some of the neurological complications that are seen in humans. Did you examine the brains of the primates in which you observed protection against the virus? And also, how did you administer the challenge? Was it administered through the nose, as is most frequently occurred in human infections?
Yeah, that's right. I don't have the data on the autopsy of the animals available to me yet, so I can't answer the first part of the question, but it's a really good question. As you know, this is a really complicated virus that appears to have pathology associated with blood vessels, with the brains. So it's a good question. I thank you for it, and I'll take it back to the group and make sure I have an answer next time I'm asked. Yes, with regard to the challenge, it was a respiratory challenge, so an inhalational challenge, yes.
As each of us asks questions, please identify also your background or your association with university or company to help help us all put it in perspective. Thank you.
So this is Bill Folk again. I'm from Mizzou.
Great. Thanks, Bill.
Nice to meet you.
Samuel?
Hi, Dr. Nettles. I'm Samuel Lam from Immunophotonics. Hi, Kelly, thank you. Thank you for that intro. We are a biotech company based in St. Louis, developing different immune modulators and adjuvants. My quick questions for you is, what do you think about different forms of antigens, protein subunits versus mRNA-based and other types of antigens? How do you feel like, is there a discrepancy, or you have a preferred form? Just how your general view on that? Thank you.
Yeah, really good question. Yeah, absolutely. There are many different techniques to approach vaccine development. And I think they all have their pros and cons. You see the mRNA or the RNA vaccines moving a little bit quicker into clinical trials. But each of these type of vaccines, I think, is going to elicit different types of immune response. Some will require multiple vaccinations. Some, the immune response may not last the same duration. And then there's also the logistical challenges of the different vaccine types. So, it will be important to understand what are the storage conditions, how are they packaged in order to really mobilize this and vaccinate all of the Americans. But having said that, I think it's too early right now in the development of the research to understand which of the vaccines might be the best. And it's probable that we'll find that certain vaccines may do a better job in certain populations. So there may be a vaccine type that may be better in elderly, or maybe better in the young. So that's to be determined. And the important thing now is that we foster all the different types of the vaccines so that we are in early next year, we're in the enviable position of being able to discern between the benefits of the vaccines, because we have seen multiple succeed.
Thank you very much.
I believe Daniel Hoff has a question.
Yes, thank you. Very nice talk, Dr. Nettles. My name is Dan Hoft. I'm the director of the Center for Vaccine Development at St. Louis University. Currently engaged in the co-VPN and starting with the Moderna vaccine. In the early days, at least, there was a lot of discussion about the actual structure of the spike protein and thinking that you might have to have it in that open conformation with the two proline mutations to get the receptor binding domain exposed to get the best neutralizing antibody response. My understanding is that in the Janssen vaccine, it's just a full-length wild-type sequence. Is there any data that, as had been predicted, and in animal models, I guess, of MERS, at least had been seen, that if you vaccinate with the closed form of the protein, that you just get binding antibodies that can actually enhance infection of cells without neutralizing the ability of the receptor binding domain to engage with the ACE2 receptor?
Yeah, very good question. You know, I think one of the things that is front and center with the FDA, as I'm sure you're aware, is that we're very careful to not induce any kind of vaccine enhanced infection. And so that is something that we are looking very closely at. In the data that we've seen so far in the preclinical model, we do not see any evidence that that we're inducing any kind of vaccine-enhanced disease. And with regard to the actual open versus closed binding site, I'm not aware of data that we have with our vaccine that I could use to answer your question. I'm sorry about that.
Well, that's reassuring. You haven't seen anything in at least the preclinical realm so far.
Right, and we're looking very closely if we're seeing a Th1 versus Th2 response, and so far it's been a Th1 response, which traditionally would be much less likely to be associated with vaccine-enhanced disease.
Thank you. We do look forward. We're signed up to also be a site for your vaccine once it starts as well. So we look forward to that.
Outstanding. Thank you.
Dr. Nettles, is your team and are the other industry players that you mentioned, perhaps as many as 100 different vaccines, are trying to go at this and getting a shot on goal, if you will, and that are they finding the number of clinical trial volunteers that they need? Is there messaging to the general population that needs to be happening from those of us with some scientific background or mantle, if you will?
Yes, in general, we're finding that there are adequate volunteers to participate. We're in our phase 1/2A clinical trial now, so we're only looking for about 1,000. With the phase 3 clinical trial, we're obviously looking for a lot more individuals, so we're hopeful that things continue to go in the right direction. The real challenge with this is using the epidemiology model to try to select places where, unfortunately, where there's a lot of coronavirus spreading in the community, and trying to predict so that when we begin to enroll in the trial, you're not past the peak of the infection rate. So in other words, we wouldn't want to be in New York City right now, who's past the peak of the infection rate. You'd want to predict where the infections are going to be in September. In order to show efficacy, unfortunately, you have to be in a place where people are becoming infected. That's the real trick. That's the hardest part of it. But when we have started to enroll in our phase 1 trial, yes, people are stepping up and enrolling. We want to make sure that continues. So yeah, we're going to give this a lot of attention.
Well, you can jump a plane to Jefferson City just as soon as you can. We've been blessed for the last 6 months with only a couple fatalities directly back to COVID, but the news the last 2 days is that a nursing home here has 39 residents that have tested positive, and 28 of the healthcare workers for that particular nursing home that has no single rooms. They're all double rooms, and suddenly it becomes real for everybody. when you have that type of direct hit. And now there's no one in Jeff City that does not know someone that has a loved one in there. And so bringing it all home.
Yeah, I'm sorry to hear that. I think you see some similar stories coming out of colleges and universities that are trying to open up, unfortunately, as well.
This is Dina Ladd with Missouri Cures. Hi, Kelly.
Hi, Dina.
Rick.
Good morning.
Good morning, Rick. That was a great presentation. So kind of to Kelly's point, as the vaccine does come out and maybe there's a limited amount, will that go to healthcare workers, frontline workers first? And also, what do you think the pricing will be on the vaccine? Thank you.
Yes. So in the beginning of the availability of vaccine, there certainly will not be a supply sufficient such that everyone can receive the vaccine. And so it's likely that there'll be recommendations that come out of the CDC, and they have a committee there called the ACIP, Advisory Committee on Immunization Practices, which will likely make some recommendation on who should receive the vaccine first. They'll probably be informed by the National Academy of Sciences, who has put together a committee who are making recommendations along these lines as well. If I had to predict, the recommendations would call for prioritization of frontline healthcare workers, which would be physicians, nurses, people in the ambulances, prioritization of individuals who are over a certain age, maybe over the age of 65. And then there are a lot of discussions about prioritization of underserved populations with regard to healthcare who are bearing an inordinate amount of burden relating to this disease. So African Americans, Hispanics, particularly in places where the population is dense. Those are the nature of the conversations that I've heard so far. But again, that would probably be a recommendation which would come out of the CDC. And you know, with the thought of opening schools, you know, are they going to include people like teachers and others now who are becoming essential healthcare workers, like the people in grocery stores and such? With regard to the pricing issue, we haven't issued a price for the J&J vaccine. I think in general, we're communicating that it's a not-for-profit price. We have a contract with the US government for the first 100 million doses of our vaccine. And I believe the government has communicated that they're committed that people would pay nothing when they receive the vaccine during this emergency authorization use period.
Dr. Nettles, where do you go for that kind of production capacity to hit hundreds of millions of doses in bioproduction? As I look around Missouri, we've got Boehringer Ingelheim up in St. Joe, and the Thermo Fisher site, which used to do the Remicade product for J&J, and you know, we're talking a handful that are in our backyard, but and certainly in light of the stated policy goals of trying to get more of this production in the United States, versus while balancing the need to get it to the patients as fast as we can once we find safe and effective.
Yeah. So one of the beneficial features of the J&J vaccine is our production capacity is high, and we're able to produce large quantities of our vaccine in a liquid media, which is beneficial. I know some of us thinking of vaccines, we might think of the old style where you had to produce the vaccines in chicken eggs, or you have to produce the vaccine on Petri dish plates of cell culture. And those types of older technologies for vaccine production require a very large footprint to produce the vaccines. Fortunately, with this type of vaccine and being able to produce it floating in liquid media, the footprint is not quite as big as the older technologies. So we partnered with two groups, which were the Emergent and Catalent companies that I mentioned earlier. One of them is in Maryland and one of them is in Indiana. And then we are partnering really around the world to find similar manufacturing capabilities to be able to produce as much of the vaccine as we think we're going to need to produce globally. Yeah, it is interesting times, certainly. So with everybody trying to produce these vaccines, production capacity is limited. So if you do have companies that are interested, you can always send me their name and I'll make sure that we have a conversation with them. And as well as production capacity is just raw materials, with being able to find glass vials and such to put the vaccines in. Just an unprecedented situation that we're in right now.
Lots of pressure on you all to knock it out of the park, and we've got folks that have really dramatically changed their business models also to lean into this on a variety of fronts, and it's that pulling together and sharing of resources is critically important for all of us to keep an eye on that. Any other questions? We've really held this group quite strongly. We've had very little drop off, Dr. Nettles. So
That's good.
they have been very interested in your presentation, and we want to thank you for that. Last call, people.
I have one more question. Well, go ahead. I already asked one. I see someone else trying.
John Buckstaff with KCS Bioanalytical Biomarker Service. Dr. Nettles, first of all, thank you for taking time today with us. Obviously, as an outsourcing service, we provide services to the pharmaceutical and biotech industries, and we've become quite familiar with the drug development process. But because of the pandemic, there's been this, shortening of that by, de-risking, for example, the companies and the way they approach the drug and the drug development. What observations have you seen, or can you comment as to, does this have any legs in the future development, how we think about drug development or vaccine development, so that companies, are getting through that risky development phases faster, or can get to the end game faster so we can get products out? What's been your take on seeing this process unfold?
Yeah, that's a great question. I think there are certainly lessons to be learned on how we can be more prepared, just like there is in the public health system, to make sure we're more prepared the next time something like this happens. I think there's a lot of lessons to be learned there. I think with specifically the development of vaccines and pharmaceuticals, I think one of the biggest lessons is developing platforms that you have flexibility, such as this adenovirus-based platform or some of the other platforms that you're seeing companies use, so that you can try to prove the ability to manufacture at scale quickly, prove the safety of the platform, and then when you have a new infection that comes along, just make a modification to a platform that you already know a lot about. I think that's a nice model. I think, though, we need to be cautious about assuming that that this is easy or sustainable. I think you see massive resources going towards bringing forward solutions at this rate at the expense of development in other issues. So, for instance, bringing forward drugs and solutions for other diseases maybe in certain instances get put on the back burner. And so I don't think that the lesson that we can necessarily learn is that now going forward, we should always move in this manner as quickly as we can. I think it's just not sustainable. but at least building the infrastructure and the flexibility into your platforms, I think that will help us be ready for the next time.
This is Dan Hoff from St. Louis again. I just have I guess, sort of an ethical question. It's not specific to your vaccine, but to all of them. The phase 3 trials are being set up to have follow-up of 2 years because, of course, one of the issues we're worried about is that the immune response may not last very long after these vaccinations if the same thing happens as after coronavirus infections. So locking placebo recipients into a trial of follow-up for 2 years and therefore potentially prevents them from getting any vaccine does become an ethical issue. So I wonder if you guys have thought about that, and I think that the interim analysis at probably at about 7 months after vaccination, if it's great news that there's a response, there's protection, and you can get at least emergency approval for use of the vaccine in mass public purposes, would it be possible to give the placebo recipients the benefit of the vaccine earlier than waiting through the whole 2 years of follow-up? We can follow the initial vaccinees to look at neutralization activity, for example, for the 2 years, just to make sure that it persists, and perhaps look at Th1 responses as well.
Yeah.
But
I think that's an outstanding question. We're working closely with the NIAID and the FDA, NIH on our design. But your point makes a lot of sense to me, and I'll take that back to our research and development group to make sure that question has been asked, because your point to me makes a lot of sense. So thank you.
Okay, thank you.
Any other questions, folks? From my vantage point, the only thing I'd like to put a plug in, Dr. Nettles, is do not have the Augusta National Golf Course in charge of deciding who gets these vaccines. I've not got my master's tickets for at least 10 years through their lottery system.
Okay, we will certainly not do that.
Okay, good.
Thank you once again for inviting us. We really appreciate the partnership with your group, and you know, as things proceed, if you would like us to come back, we'd certainly be happy to.
Well, you have an open invitation. I've even got 1 or 2 of the attendees that have asked me to see if you might be willing to speak to one of their upper level courses that they teach. So we'll get those forwarded on to you. We thank everybody for inside the MOBIO family for participating in this month's briefing. And please be thinking about other valuable content that needs to be discussed by our group, and we flex into that. And we miss seeing each of you in person, but we hopefully, with Dr. Nettles' success, we'll get back to that very soon. So everyone have a great day, and I thank you for joining on behalf of the MOBIO Board of Directors. Thanks so much.